Hello students, welcome to Trunkie Academy. Okay, again we are today we have met to study another topic. Okay, and since we were studying about um, the sex-linked inheritance and some disorders related with sex-linked inheritance and about the crisscross inheritance, we also studied. And now today I have chosen a very important topic with regard to or with respect to the exam. Okay, and the question is many students they are confused about this, and the questions are asked based on this topic, and the topic is the DNA fingerprinting. Okay, so first of all, I'll give you some brief idea, and since why this is very important, because what I have found in past years in the questions asked in the NEET examination is that the questions are asked from the some kind of biotechnology or principles of biotechnology or DNA fingerprinting or in human from the human physiology or from the diversity in living world. Okay, so this being one of the important topics, okay, you have to listen very carefully, and I'll tell you in a very interesting manner, and you'll enjoy your learning and you'll understand the topic very clearly. So first of all, we have to start with the DNA fingerprinting. Now, a little bit of a brief idea I'll give you that when we say DNA fingerprinting, and this word and this term you have heard or in previously also maybe while watching the movies in the cinemas and or maybe in the news. Okay, somebody has murdered somebody, or there is some kind of investigation going on. So DNA fingerprinting helps to find out the murderer, or sometimes when there is to confirm the paternity of some child. This DNA fingerprinting is also a very helpful tool. Okay, so DNA fingerprinting means we'll see in detail a little later, but just a brief idea I'll give that. There is some kind of pattern, a sequence in our DNA. Okay, so that sequence we want to match. Now you see, everybody is different. Looks not. It may be a little similar, but it has got unique. It's of its own. Okay, so you can say the human races, or even in the if you go to different parts of the world, okay, or even the brothers, okay, and even the twins also, okay, there is some kind of differences. Okay, so you see that what is there that is making us unique from one another. So your rainbow lies within. Okay, that means see, yeah, it's our genetic material. Yes, so every person is unique of its own, and this is the DNA which makes the difference between create a difference between one another. Okay, in human beings, this is the making us unique from one another. DNA. Okay, it is the DNA that makes us different or unique from one another. Okay, so like cookbook. DNA acts as our cookbook to tell how to grow and develop. Okay, there's a blueprint. Okay, so there's a blueprint for construction of the building. So accordingly, the design and the number of floors will be constructed. Okay, depending on the blueprint. So DNA also contains the blueprint how we will develop and how we grow. Okay, what will be the size of the nose? What will be the the length of the pinna? Okay, whether it is joined or it's free ear lobes. Okay, if the hairs are, it is a little spiral, or it is straight, okay, or curly or straight, okay, or the color of the skin, or so many different things. The thousand stuff, okay, features are there. They're directed by the DNA for a particular person. Okay, so this DNA is present in the chromosome. Okay, so 99% of the chromosome is similar to everybody. Okay, only 1% or 0.1%, 99.9% of our DNA we share with one another. So you just imagine, 99.9% of our DNA is similar to everybody. Okay, only 0.1% of our DNA is different, and this only 0.1% of our DNA makes a difference from one another. Just imagine. So that means only 0.1% of DNA is responsible for making difference from each other. Okay, so just remember this. Okay, so a very small amount of difference that makes us unique. So just in the DNA, it's a very small amount, but this but makes us unique from other persons. Okay, so this means that we all share most of the information written in our cookbooks or your DNA, or we can say our the blueprint, but there are some variations in the recipes. Okay, that means 0.1%. So this is the uh, thus we are unique from one another due to that 0.1% of the DNA. 99.9% is same in everybody. Now in a population, every person shows unique sequences. Okay, so variable number of tandem repeats. This is called variable number of tandem repeats. Now what is this tandem repeats? I will see. Okay, in DNA there is 0.1%. Okay, there is a kind of repetitive DNA we call this satellite DNA. Satellite DNA. And this satellite DNA, there is the mini satellite DNA and microsatellite DNA. Microsatellite. Okay, and this macro and mini. And in case of this microsatellite DNA, there is the VNTR. And with the help of this, we do this DNA fingerprinting, VNTR, variable number of tandem repeats. So we will see what is this. It is a sequence of bases of DNA that is repeated at a specific chromosomal locus. So in the chromosomal, okay, the DNA are arranged, okay, in the present, and the number of genes or DNA, okay, the base is repeated, okay, in sequence. Okay, how it is repeated? That is called the VNTR, variable number. So it is a sequence of bases. The bases in DNA that is repeated at a specific chromosomal locus, in a specific chromosomal locus position, in position, in a specific chromosomal position locus. Okay. So like here VNTR regions. Okay. So VNTR region is very small, and from there we need to highly polymorphic. Okay. And it was discovered by the A.R. Wyman, okay, and R. White in 1980. Okay, the VNTR. Now, length of the regions having VNTR is different in each individual. Okay, so in every individual, length the VNTR, variable number of tandem repeats, will go on for how long? Across the length of at what extent, that is different in different individual. Okay, so key factor is the DNA fingerprinting, means name has been kept because the fingerprint is different in different people. So this there's a sequence. Okay, the VNTR is also different in different people. So that's the name was like DNA fingerprints. It's not the normal fingerprint; it's a DNA fingerprinting. So over 99% of the 3 billion nucleotide pairs in human DNA are identical among all individuals. So okay. So 99. Now, no two people other than identical twins have exactly same sequence of bases in their DNA. Okay, except the twins. In all other individuals in this world will have a different set of DNA sequence. Okay, in the DNA. Not exactly same, it will be different. So restriction, fragment, length, polymorphism are characteristics to every person's DNA. RFLPs are pronounced as reflex. Okay, reflex are unique or is a characteristics to every person. Okay, restriction, fragment, length, polymorphism means we'll see that also. It means that are the called variable number tandem repeat. This restriction, fragment, reflex, RFLP polymorphism are variable number of tandem repeats, but that in 0.1%, in 0.1%, okay, how the bases are arranged in sequence. Okay, that is the forms the base for the DNA fingerprinting, and that is called RFLP and variable number of tandem repeats. The variable number of tandem repeats are of 2% genetically show variation. RFLP. So try to. I'm repeating repeating once again, and please try to understand and listen very carefully. In 0.1, 99.9% is similar. The bases are same. The DNA is part of the DNA is same in everybody, but only 0.1% is variable. And within this 0.1%, okay, we have got some sequence arranged. The bases are arranged in a particular pattern. Okay, that arrangement of the bases, the sequence of the arrangement of the bases will be different in all different persons. Okay, so this is the the VNTR, the repeats, the repetition of the. For example, there is a example of the DNA sequence. So there's A, G, A, C. Okay, this is the and this will be keep on repeating. Okay, after in what sequence and how many frequency. Okay, it's repeating. Okay, so that is the VNTR or RFLP. Okay, polymorphism means different is form. Okay, in different persons. So what happens? DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called repetitive DNA. So DNA is repetitive. Okay, that forms the basis because in this sequence a small stretch of DNA is repeated many times. Okay, so that is the VNTR, and the sequences show high degree of polymorphism and form the basis of DNA fingerprint. So that only part which is repeating and forming, that is means because in this sequence a small stretch is repeated many times, and this repetition is different in different people. Okay, and this repetition is so the polymorphism means it is different form and a different arrangement is shown. Okay, in different person and in all the persons. So that forms the basis of the DNA fingerprinting. There are bits of chromosomes that can be cut by restriction endonuclease. Restriction endonuclease is an enzyme used in the biotechnological tools to cut the chromosomes. Okay, and to study about the DNA and the chromosomes. Okay, so the fundamental technique involved in the DNA fingerprinting was pioneered and perfected by Jeffries of Great Britain. Okay, so Jeffries, he is the scientist who actually discovered this technique. Okay, of Great Britain. Now, Jeffries observed that the DNA pertaining to myoglobin of muscles contains many segments that vary in size and composition from one person to another. When he was studying about this DNA and the sequence of the bases in the DNA, so he observed that the genes pertaining to myoglobin of muscles, okay, or the genes that is present in the DNA of the myoglobin muscle contains many segments that vary in size and composition from one person to another. So this clicked in his mind, and this formed the base for the discovery of this DNA fingerprinting. For example, in the following hypothetical example, nucleotide base sequence, there are 6 tandem repeats of 16 base each. Count the first 16 and note how they are repeated. First, you see. Okay, so here is the repetition. So you can see. See here is the repeat. It is about the GC TAAGA. This is a repetition. So GACT. So this is repetition. GACT is repeated. Such clusters, 10 to 100 nucleotides, okay, are called mini satellites. Okay. So GACT, guanine, adenine, cytosine, and thymine, these are in the repeated. Okay. So this will form 10 to 100 times nucleotides are called mini satellites, where there will be the repetition from 10 to 100 times nucleotides. Then it is a mini. Now such tandem repeats are characteristics of every person's DNA. Okay. Now the VNTR of two persons differ in number. Now another point also I would like to tell you, there is another in the satellite DNA, then micro and mini. Okay, apart from the mini, there is micro also. Okay, and this forms and there is we have got the VNTR, and it forms the basis. VNTR are two types. Two persons differ in number of tandem repeats or the sequence of bases. Such changes are caused due to mutation and gene recombination. Okay, so VNTR of two persons differ in number of tandem repeats. So that sequence, like GATC, then GATC, how many different and after how they are frequently occurring and how they are the sequence they are arranged. Okay, they are repeated. Okay, that forms the basis, and this is called the VNTR. Okay, very number of tandem repeats, and that forms the basis for DNA fingerprinting, and that's repetition. That sequence arrangement is different in all the individual. Okay, such changes are caused due to mutation and gene recombination. Why such changes are there? Why the repetition is different in different individuals? Why there is a change in the repetition and the arrangement and sequence is different? Okay, sequence will be maybe the same, and then the repetition may be the different. Okay, so why does it change? This is because the recombination. The people are when the newborn is the people are getting married, and then when there is a reproduction takes place. Okay, there is mixing of the genes. Okay, recombination takes place, and also due to mutations. Okay, the change in the gene due to mutation. Another example: a child might inherit chromosomes with 6 tandem repeats from mother, and the same tandem repeats 4 times from the father is in a homologous chromosome. So the same tandem repeat will be inherited from the mother 6 times. Okay, 6 tandem repeats will be from the mother and 4 from the father in a homologous chromosome of the child. So it means that half of the VNTR alleles of the child resemble those of the mother and other half are those of the father. So this is the heterozygous condition with reference to VNTR alleles. So this is the heterozygous conditions. Okay. So now the tandem repeats serve as the basis of technique for DNA fingerprinting. Now what is DNA fingerprinting? A technique used by scientists to distinguish between individuals of same species using DNA as a sample. Okay. So this technique is used to distinguish between 2 individuals. Okay, by using the DNA as a sample, by studying the tandem repeats. Okay, by studying that DNA, the DNA basis repetition. Okay, so this is used to distinguish between the 2 individuals. In 1984, Alex Jeffries from England, he was the discoverer and the scientist who discovered this technique of DNA fingerprinting. Okay. Now, DNA fingerprinting is also known as DNA typing or genetic fingerprinting is a method for identifying individuals for the particular structure of their DNA. Okay, that is the repetition of the DNA fingerprinting is used for many purposes, particularly detecting the paternity, maternity testing, and for forensic work like the to know the criminal or murderer. Okay, so in different situations, DNA can be obtained from whole blood. Okay, or from the semen, or from the vaginal fluid, root hair follicles, almost any tissues. Okay, almost any tissues like from the skin, muscles, or even from the bones that have been buried for a long time. Okay, for fingerprinting. If it is buried under the earth, okay, and it is decomposing also, from there also DNA can be obtained. Okay. So now this is the whole blood, and this is the semen, and this is about the hairs. Forensic, and this is about the any tissue from the skin or muscles and the bones, etc. Okay. Now, DNA fingerprint is a technique by which the DNA of an individual can be compared with that found in the sampler or another individual suspect in a crime. For example, we have got 2 kinds of blood and there has been a murder of a person and it's lying on the floor and there is a blood. But we found 2 different samples. So obviously that one blood sample is from the person who is already dead, okay, and one blood sample that may be suspected as it may be of the criminal or murderer, okay. So that we need to check with the person we the suspect. So we take out the sample of the blood from that person which is suspected and will match with the blood which was found in the crime scene. Okay. So ATGCR it is an N2 basis or sugars or ions or it is an nitrogenous basis. Okay. The nitrogenous bases are present in the what we call this in the DNA. Okay. Now, DNA fingerprinting protocol advantages. Okay. So how this is done and advantage. DNA fingerprinting, it is done with the help of gel electrophoresis and southern blotting. Okay, that will study in detail. The technique is carried out as follows. First, it is DNA isolation. So first we have to isolate that, take out the DNA. We need to take out the DNA from the samples like blood, bone, or skin, or muscles, or from the hair follicles or hairs, etc. Okay, so we need to isolate, take out, isolation. Okay, DNA obtained from blood, semen, hair root, sample, etc. Okay. So now how to obtain this? So we need to centrifuge. Okay, sample 1 is. So we need to centrifuge. If DNA is obtained is very less in quantity than it is amplified using PCR. So because we need to number more copies. Okay, large, a little large. It is very small in quantity, then we need to amplify. Make more copies of that to study easily, okay, by PCR, polymerase chain reactions, to amplify. And if the sample is more, then we need to isolate by centrifugation, okay. So polymerase chain reaction helps in amplifying or increasing the samples, okay, of the DNA. Now DNA fragmentation. The next step is the DNA, the fragmenting the DNA, by cutting small pieces, okay, by restriction endonuclease. So a kind of enzyme is there which is called restriction endonuclease that acts as molecular scissors, also called regarded as the molecular scissors that cut the DNA at specific sites and it brings into smaller pieces. Okay, that is by the many fragments, and it brings in fragment, and many fragments are obtained having variable length. Okay, so many fragments are formed after applying this restriction endonuclease because they cut into small pieces or fragments, and many fragments are obtained thus. Okay, so our restriction fragment length polymerism means that I told you earlier that there is different now after that we in PCR first is the annealing, then how the extension. You see, this is a process. Okay, when we find the denaturing DNA, the DNA on the gel is denatured using alkaline chemicals or by heating. So what we are doing is we are using alkaline solution or alkaline chemicals, okay, to denature. Okay, denaturing means separating and splitting of the double helix. So here you can see. So what we are doing is extension 72 degrees Celsius or then 94 degrees Celsius, and then we are denaturing, or we are applying some alkaline solution to separate in splitting the DNA helix, which is the helix which is there in that, because DNA is the double stranded and it is in a form of helix. Okay, so to separate that strand, the single strand from double strand, to make it single stranded, okay, to separate from each other, that is the denaturing the DNA, or splitting or separating into single strands by breaking the hydrogen bonds, because the double helix, okay, is formed. Double helix formed of the two strands is formed by the hydrogen bond between the two strands. Okay, the hydrogen bonds, breaking the hydrogen bonds between the two strands. Okay, and then separating into single strands. That is the denaturing, or it is the splitting. Okay, so this is done by heating and using the alkaline chemicals. Okay, or alkaline solution. Now the fragments are then separated by agarose gel electrophoresis. This is a kind of technique where some agarose gel is placed, and then with the help of micropipette, the samples are placed in the regions. Okay, so this is a solution, agarose gel, and you can see here, see, there's the solutions. Okay, and then yes, you can see agarose gel. Okay, and DNA sample is placed. Okay, in well cut gel. Then the gel is cut in certain region to place the DNA samples with the help of micropipette. Okay, and then it is this agarose gel is connected with the anode and cathode. Okay, so that there will be the movement of the molecules. Okay, the voltage supply, then the negative electrode and the positive electrode, it will connected to, and then what will happen? Then the movement of these fragments depends on their length. Then filled samples are there, isn't it? So after slowly you will find that there is a movement of the fragments. The movement of the fragment takes place. So after doing the fragments with the help of the restriction endonuclease, so what we are doing is we are applying. So filled with the samples, and then there is the what we call this, the movement of the, you see, then the movement. Okay, that is, this results in the formation of bands. Okay, so this particular band is different in DNA collected by different persons from different persons. The bands are then blotted on nylon membrane or nitrocellulose membrane. So what happens whenever these bands are formed? To get a clear picture, okay, to picture of these bands, okay, one nitrocellulose membrane is placed on that, okay, so that the bands that has been formed on the agarose gel, that will be that will form one image, that will be the picture will come on the nylon membrane or nitrocellulose membrane. Okay, so this is called the southern blotting. Okay, so one nylon membrane is placed. Okay, and then what will happen? Single stranded DNA gets embedded to the nylon membrane. Okay, so that which the fragments of the DNA which has formed the bands in the agarose gel during the gel electrophoresis, okay, that will be embedded to the nylon membrane when we place on the agarose gel. Okay, when you place the nylon membrane on the agarose gel, the DNA fragments, which is in the form the bends on the agarose gel, okay, after the gel electrophoresis, that will get embedded on the nylon membrane or nitrocellulose membrane, and then we can absorb easily. So as the towels draw the moisture DNA, the DNA strands are transferred onto the nylon membrane by capillary action. Okay, and you know capillary action is very thin and thin tubular structure it draw in the water. Okay, and the chemical, the liquid. Similarly, here also a nitrocellulose membrane or nylon membrane acts as the capillary the capillary reactions. The DNA strands are transferred to the nylon membrane. Okay, now this process is called blotting, and more precise, it's called Southern blotting after the name of the inventor E.M. Southern. So the scientist who discovered this technique, his name was E.M. Southern. The blotting means like with the help of blotting paper, we are using the absorption of the water. Okay, with the help of the blotting paper. The blotting paper has got that capillary action. Blotting papers are thick and has got some air stresses, and that will absorb the water. So blotting. So here we are also a kind of a blotting. We are allowing the DNA fragments to be absorbed, to be embedded in the nitrocellulose membrane. So it's a kind of blotting, okay, similar to blotting. So it's called blotting. And why Southern blotting? Because the scientist who discovered this technique, his name was E.M. Southern. So the name is Southern blotting. Okay. Now hybridization. The bands are flooded with a single stranded radioactive probes. What will happen once we are getting the bands? Okay, that means that when these fragments of DNA which has formed, okay, due to the DNA fragments and during the gel electrophoresis, which are now embedded on the nylon membrane. Okay, now what we are doing, we are now for example, if there is the band of the DNA like G-T, okay, so what will happen there? Here will be the complementary, the other complementary will be the T, C and A, okay, and the G. So if there is this band has got AGTC, or so complementary will be the TCAG. So what we'll do with that, we'll provide the band with the single stranded radioactive probes, okay, and we'll provide with the DNA strand, okay, single stranded DNA with radioactive atoms and radioactive substances, okay, so that it will shine on the radioactive plate. It will radiate and shine on the radioactive plate, and we will get the actual. Now finally then we can get that. So what will happen? Till now we have not got the clear picture of this band. Okay, we know that during the gel electrophoresis, the bands has formed. Okay, and then these bands has transferred the DNA fragments has transferred to the nylon membrane. Okay, and then we are again being flooded with the single stranded radioactive probes, which are the two known. Okay, to know the difference, to find out how it is going on, okay, and how it is found in the next step. Okay, so what we are doing is we are providing the band in the nylon membranes with a single stranded DNA with radioactive substances, labeled with radioactive substances. Okay, so for example, probes get complemented to complementary DNA sequence. As I told you, if this is the AGTC, okay, and then we will provide a TCAG with radioactive substance. So that the bases formed with the radioactive substance. So that will, since it will radiate and it will shine on the radioactive plate, and then we will get the plate. Now the probe attaches by base pairing to those restriction fragments that are complementary to its sequence. So what will happen? So when we are providing, okay, we are flooding with the radioactive probes, okay, the base pairs, okay, that will join and go to its complementary base pairs, and it will join and form the sequence, okay. And what will happen then? The probes can be prepared by using either fluorescent substances or radioactive isotopes. For example, in the base, nitrogenous base, substances. Okay, the different elements, instead of using the normal elements, okay, we are using the radioactive elements. Okay, so that is going to be radiate, and that is will be there will be the shining, and there will be the kind of image on the radioactive plate. Okay, now excess probes are washed off. Now once there has been the a formation of complementary strand, okay, the fragment which is present in the nodal membrane, that will take the complementary part from the one which was flooded there, and then the extra are washed off. Okay. So now what will happen? The photographic. The next step is the photographic. Then we place one. The nodal membrane is kept in contact with X-ray film. Okay, in contact with X-ray film, and this will the DNA bands due to radioactive probe give photographic image on X-ray film. As I told you, because since the DNA which has the radioactive elements, okay, and that has went to form the complementary strand which was already there in the nodal membrane. So now that will radiate, and that will shine on the X-ray film, okay, and that will then it will form an image on the photographic film on X-ray film, okay, photographic image. The thick and thin dark bands form a pattern of bars which constitute a genetic fingerprint. Okay, now this will be ready for matching with the. This is how the developed. This is how the developed X-ray films look like. Okay, like here. So this is called audiogram. Autoradiograph. Autoradiograph means autoradiography, because that autoradiography means when the DNA bases which containing the bases with the radioactive substances, that is going to form the image. Okay, due to its radiation. Due to radiation, this comes out of the radioactive substance which is there in the nitrogenous base. Okay, so what will happen? DNA fingerprinting. Parental dispute, catching criminals, these are used. Genomics, forensic science. Okay. Isolate chromosomal DNA from a sample of cells. A sample of cell, one cell obtained from the blood, hair, roots, or semen. Okay, and then we extract. Okay, the DNA. Cut the DNA with restriction enzyme. We isolate. Okay, and then we isolate the DNA. Cut it with help of. Then we do the gel electrophoresis to form the bands, and now after that we are solvent blotting, transfer before this transfer band to the nitrocellulose, okay, because this is then embedded to the nitrocellulose, okay, and stake of paper towel is there, the nitrocellulose, okay, then filter paper with salt is there, solution gel. Then we liquid containing radioactive probes. Then we are cDNA, RNA, or cloned genomics DNA. Then we are flooding with the bases with radioactive elements, okay, and denature the DNA and add the probe. Oh, sorry. Before that, we need to denaturate the DNA and add the probe and allow the probe to hybridize and wash away the excess. What we are doing, we are providing with the DNA fragments with maybe the complementary to the one that is present in the nitrocellulose membrane. Okay, so what we are doing, the DNA complementary DNA which we are providing, okay, or we are flooding with, that we are labeling with the radioactive element. So that is called radioactive probe. So we are that will hybridize, allow probe to hybridize and wash, and then we wash away the excess probe, and radioactive probe hybridize to nitrocellulose filter, and they form the complementary base with the one that is present in the nitrocellulose membrane. Okay, the hybridized nitrocellulose washed and autoradiograph, expose the filter to photographic film and compare the results. So this is how we get the bands, and we compare with the two different samples, sample 1 and sample 2. Okay. Now a given person can never have a VNTR which his parents do not have. Okay. So please remember this: a given person never have a VNTR, okay, which parents do not have. So if parents do not have that VNTR, then the son or the children do not have that VNTR. Okay. Now, so obtaining hybrid with radioactive probe and matching DNAs of different members of the family with biological children and adopted children gives us an idea how DNA fingerprints helps identification of paternity, maternity by studying the DNA fingerprints members of the family, biological and non-biological relationship. Okay, so if VNTR or is not matching, okay, means that there is no relationship between the father and the son or the daughter. Okay, if somebody is claiming a child, father of that particular child, so the VNTR should match definitely. If it is not matching, means he is not the father. Okay, so illustration given below are the VNTR patterns for Mrs. Rose Blue, Mr. Rao Yellow, and D1. Her biological father, D2, child of Mr. Rose, and her father husband, and S from Mr. Rao's biological son. Okay, not biologically related. Okay, the illustration is not given here. Not biologically related, expired, and marked in light and dark. Picture is not given. So this question is incomplete. Now here you will see mom, dad, okay, and then the daughter 1, daughter 2, and son 1 and son 2. Okay, now you can see the dad has some kind of pattern. Okay, so that has been taken from the mother. You can just in son 2, you can see son 2 in the son number 2, okay, S2, you can see the green one. Okay, the one pattern has been taken from the mother, and that has been taken from the last two below. This is the taken from this pattern. This repetition is taken from the mother, and these two taken from the father. Okay. Now here in this case, here this is taken from the mother. This is taken. This is taken from the father. Okay, this is from the mother, and this is from the father, and this is from the mother again. Okay, so it is from the M. This is from the M. This is also from the mother, and this is from the father and from the father. Here also it is from the mother, and here it is from the father, and this is also from the father. Okay, so here also you can see this is from the mother. Okay, here it is from the. This is also from the mother, and here it is from the father, and this is mathematics. So here again you can see this is with the mother. Okay, so there is some similarity with the mother and the father. This is pattern. So we match likewise. Okay, so this then will not match with the DNA from the other persons. Okay, so here it is with the mother. Okay, here yes, no sir. Yeah, and then here it is the, here this is from the, sorry, this is from the mother. Oh, no, this is not from the, here it is from the father, and it is from the mother again. Here it is from the mother, no, new pattern, and here it is the yeah, this pattern is not matching, you see, because he is dad and he is mom. They are not integrated. So since they carry because their DNA has come from their parents, so it's not matching, you see. This is not matching, isn't it? So this pattern is not matching with this pattern. Okay? But son and the daughters are somehow matching with the pattern and the sequence, or you can say the repetition of the DNA of the mom and dad. Okay? So this is how the DNA fingerprint, okay, is done. So selection of victim's DNA, okay, of 24 repeats of GT, 16 repeats of ATGG, and 66 repeats of ATT. So this is how, okay, bases, nitrogenous bases, repetition. So how they are repeated, okay, in how many times, okay, they are repeating, okay, and then there is a gap, and then how many times they are repeating. That the repeating pattern is very important, repeating pattern, the repetitive DNA, okay, so 24 repeats, okay, then 16 repeats of ATGG, and 66 repeats of ATT. So if this is matching with another sample of DNA, definitely it is the same person, or sorry, that means this is a sample of from that particular person. Okay, so there are three regions in the repetitive DNA. Section of suspect DNA. And what happens? Then there is the 49 repeats of GT, 16 repeats of ATGG, and 51 repeats of ATT. The same three regions of repetitive DNA are present here, but some include different numbers of repeats. Now let's compare to this center. Okay, select section of DNA from crime scene here. Okay, so here we see 49 repeats of GT, 16 repeats of ATGG, 51 repeats of ATT. Length of the repetitive DNA sequence matched the length in the suspect's DNA. So DNA found in the crime scene belongs to the suspect. Okay, here in this case. Okay, now application of DNA fingerprint, conservation of wildlife, protection of endangered species by knowing the DNA. Okay, by maintaining the DNA records for identification of tissues of the dead endangered organisms. Okay, this is it. Taxonomical application of study the phylogeny. How from which animals has evolved from which animal, or which plant has evolved from which plant. The pre-existing plant, the animals and plants have evolved from the pre-existing organisms. Okay, and what was that particular organism, or maybe the relationship between the organisms. Okay, like if we have evolved from the monkeys, how are we related? What is the percentage of similarities between the DNA of the monkey and the human beings? Okay, there's so many things. That's called taxonomical application. Then the pedigree analysis. Okay, inheritance pattern of gene through generations. How the different features are inherited from our grandfathers to the mother, and then to the son and the grandson and the great grandson. Okay, so anthropological studies about the different human races and the population and the origin and migration of human population. Medical legal cases like establishing paternity or maternity more accurately, and forensic analysis for the to identify the suspect in a crime. Okay, and now we'll go some questions. Genome is technique for DNA sequencing or complete set of genes present in an. This is very simple. It's a complete set of genes present in an organism. Okay, it's not particular segment. It's a complete set of genes. Okay, so the complete set of genes present in the organism. Okay, in all chromosomes. So that is the genome. Human Genome Project was initiated by U.S. Department of Agriculture, Energy, Science and Technology and Health. It is the energy. So this was all about the what we call this DNA fingerprinting. And very brief, I will recap and tell you that there are in the chromosomes there are DNA is present, and this DNA are made of the nitrogenous base pairs. Okay, means they are single strand and they are joined by hydrogen bond to form the double helix. Okay, double strand. Now, what happens in the chromosomes when the DNA are present? This DNA is actually the blueprint for the to give out and to express the different features in the progenies in the offspring. Okay. Now, the 99.9% of the DNA is similar to in everybody. Only 0.1% is different. Okay. That makes us unique from each other. Okay. Now, with the help of the 0.1%, we are actually able to do this bigger DNA fingerprinting. And the DNA, in that 0.1% of the DNA, there is the VNTR. There is satellite DNA. That's called satellite. In satellite DNA, there is micro and mini satellite. And microsatellite is actually the forms the VNTR, variable number of tandem repeats, also called RFLP. I discussed it. Okay. So that is the variable. And what happens when we are for DNA fingerprint, when taking out the sample from the blood, we are extracting or isolating the DNA. Okay, if it is very less in quantity, but we are doing the PCR, polymerase chain reaction, and if it is simple, then we are centrifuging. The higher, the heavy one goes down, settle down in the centrifugation, the lighter one remains and above level. So then we, after isolation, we are cutting with the help of mechanical scissors the restriction endonuclease. So many fragments are formed. Try to listen this, recap what I am telling you. So many fragments are formed, okay, and that fragments are used for gel electrophoresis, okay, and during this gel electrophoresis, when the electron connected to the electric current is passed, so what will happen? That it will move, okay, into the agarose gel. Gel electrophoresis means there is a kind of notches in the cut areas where with the help of micropipettes the DNA samples are placed, and then through that the gel, okay, to the gel, Okay, and that agarose gel, these DNA fragments will move, okay, when there is a flow of current, and they move depending on the length, okay, and after they form the bands, but which we are not able to understand clearly. Then a nitrocellular membrane is placed over, then that DNA comes embedded in the nitrocellular membrane, and after this embedding, that is called the southern blotting. Okay, and after southern blotting, what we are doing, we are flooding, and we are allowing to form the complementary strand. When we are giving the DNA strand, what is it? That is labeled with the radioactive element. Radioactive element in the nitrogenous base that will give some kind of radiation, and that is reflected on the photographic film, and then the band is absorbed in the photographic film, and that band, with the help of that photographic film, the band that is formed on the photographic film, we are able to compare with the another sample, okay, formed by another sample. So when it is matching, then we are able to conclude whether this is the criminal or whether this is the father of that particular child or not. In this way, DNA fingerprint is done. So go through your text, and it's very easy. Now after this class, we'll be sending you the daily practice paper, daily practice problem. Okay, like every day we are sending. Today also we'll be sending, and as you know, then you have to click on the resource link, and then you click on the writing material. So if you click on the writing material, you will get some the written DPP, and click on the DPP, you will find there is some set of questions, and you go through that question, answer those questions, and send back to us for evaluation. Okay. So thank you for joining today and listening, and I hope you enjoyed your learning and you understood this DNA fingerprinting. Many questions are asked, okay, based on this, okay, VNTR, RFLP, and the gel electrophoresis, agarose gel is used, nitrocellular membrane, and the restriction endonucleases, and all these things are asked in the examinations. Hope you've understood and you've enjoyed the learning. Thank you for joining today. And again tomorrow we will meet at the same time with another new topic. So till then, goodbye and thank you for joining.